this computer. Okay, here we go. Filariasis or elephantiasis is today's topic. And this is a great topic. I like to sort of start the medical entomology class with this topic because around this topic is sort of like the foundations of the history of medical entomology. And this was sort of like one of the first questions that was approached and addressed by these early scientists that we would call the fathers of medical entomology. So this is a really fun topic because it goes through the history of medical entomology, vector biology, and it's also just a really interesting disease, although of course it's a serious disease that has lots of impacts, negative impacts on people. So what is filariasis? Or I've heard people pronounce it differently, like filariasis, filariasis, however you want to say it. What is it? It's essentially a human body gets infected with nematodes. Okay. And nematodes are kind of like worms. They have a cuticle, so they have a shell. They molt. In that sense, they're kind of like insects, but they're essentially just a tube. Okay. They're essentially like tubes that reproduce. And that's quite important, the fact that they mate. They're sexual organisms and they reproduce, and that comes, you'll see why that's important in their life cycle. So they're essentially sexual tubes that crawl through human bodies. If you see, there's many different kinds of nematodes. Filarial nematodes, those are the ones sort of known to cause pathogenesis in humans. So these cause diseases, and there's many other nematodes, but the filarial ones are the ones that cause pathogenesis in humans. The order, if you want to know, is Spiroridea. Superfamily Filaroidea, and that's sort of what I'm talking about here. These are worms that cause disease in humans. They can have different morphology. So here you're kind of looking at like a generalized nematode structure, a tube with a head, a butt, and reproductive organs. They don't always look sort of like this. I mean, they could be like spaghetti-like things. They could be very elongated, or they could be very flat and short. So there's different kinds of morphologies to nematodes, but essentially you just want to remember they're like sexual tubes that reproduce. Okay, let's talk about the biology of these worms. There are young worms and old worms. So we call the young baby worms Microfilaria. These are like the tiny, tiny, tiny worms. And then there are adults, okay, which are, they're the ones that mate, and they obviously give rise and lay eggs, which give birth to or hatch. The Microfilaria hatch out of the eggs laid by the adults. And there's sexual dimorphism. So I think this is a female here. Here's a male. Usually females are larger. So this picture is not Microfilaria versus adults. The Microfilaria are very, very, very tiny. And some are sheathed and unsheathed. A sheath is sort of this, and it comes from the shell of the egg. So the actual sheath, quote unquote sheath, in the Microfilaria is actually the egg. It kind of like sticks with the nematode and causes immunological responses in humans. Anything else? The Microfilaria, typically, we're going to talk, I think, about 3 nematode-based diseases. It's usually the Microfilaria that cause the major pathology. But the problem is, essentially the source of the problem is that your body is usually infected with some of these adults. And the adults sort of seed your body with these Microfilaria. And these microfilaria are so small that they can like crawl through, they swim through like your skin. So if you know about biological organisms, they're made up of cells. Perhaps these are bad because these are square looking cells, but the cells bathe in extracellular fluid that comes from if there's like blood, right? If there's like a capillary or an artery, the blood vessels feed sort of nutrients into the extracellular fluid, which diffuses and feeds the cells. And the microfilaria can crawl around and swim around in these regions. And you can imagine as they do that, they disrupt structures and cause all kinds of problems. And once they die, we'll talk about this more in detail, once they die, their bodies are cycled into your lymph and they accumulate, and that's what causes the elephantiasis. But we'll get to that in a little bit. So just remember that it's usually the microfilaria that are really sort of causing the pathology. Okay, so let's talk about some of the history of this nematology and vector biology. There is this person, Otto Wucherer. He has a genus of these worms named after him, Wuchereria. We'll talk about more about those. But this person was famous because he would take patients like the patients, they have like this obvious like elephantiasis like phenotype. And he would take them and he would collect their urine. So we would collect urine, and then he would just look at the urine under a microscope, and he was able to see the microfilaria, the worms in the urine. So this guy is the first person to sort of identify worms in the urine, and he sort of suggests that these worms are probably the cause of that phenotype, that elephantiasis pathology. Now, there was another person independently before 1865, 2 years before, a French person named Jean-Nicolas de Marquet, who identified microfilaria in hydrocele. Hydrocele is this strange liquid that is in the scrotum in the testes. It surrounds the testes. So this French person saw kind of the same strategy, saw worms in the hydrocele. But Wucherer, am I saying that? Wucherer, he's the person who sort of like gets credit. But perhaps there were multiple discoveries of these microfilaria in humans. But the point you want to remember that that Wucherer accomplished, is he saw these in humans. Okay, so in like human, in this case, what was it again? In the urine. So different people get credit for finding these worms in different parts of the body, and you'll see that becomes a little bit more important as we start to figure out, as humans start to figure out the pathology of how this happens. Okay. Next very, very famous person. You want to remember this person. This is the father of vector biology. Or you could say the father of medical entomology. He was a Scottish doctor, okay? And he was born during the time of the British Empire. So the British Empire, Britain is an island, right? They had control of many sort of like islands and parts of the world. And they would send their doctors to sort of learn about different diseases in the world. And this guy's sort of hobby was tropical medicine. So he spent time in, I think, both China and India, and many of his observations were from India. And his sort of hobby was studying dog heartworm. He worked at a hospital in tropical regions. Again, studied dog heartworm. We'll talk a little bit about that at the end. A different kind, different genus, Filaria immitis. And he had a big hypothesis that elephantiasis was sort of like spread around by bad contaminated water. So just so you know, let me finish typing this. This time in history, this is like, people at this time did not know how diseases had spread. We knew almost nothing about the life cycles of these diseases. We knew nothing about the pathogens. This is all before those discoveries were sort of made. So just keep that in mind as we talk about these discoveries. Okay, so Patrick Manson, okay, here's a fun quote from him. I like this quote a lot. I live in an out of the world place away from libraries, out of the run of what's going on. So I don't know the value of my work or if it has been done before or better. So this guy was kind of like a recluse. He kind of like isolated himself. He wasn't a person who read a lot of the sort of common day literature. And his philosophy was, let me try to just think of my own ideas and let me follow empiricism, make my own tests, do my own experiments, follow the scientific method, and at least like the small knowledge that I have is derived from empirical experiments. That was very much sort of his philosophy, and it's enumerated in this quote. So I sort of like, this guy has a lot of good quotes. And he contributed sort of what I call his big four contributions. So you will want to remember these. I usually put these on the test. I usually just kind of say like, what were the big four contributions of Patrick Manson? His first sort of big contribution is that he proposed that the mosquito, okay, so let me go through the logic of this first. So what he would do is he would take a patient who had elephantiasis. He would extract blood, okay? And he would look at the blood under the microscope, and in the blood, right, were millions and millions and millions and millions of microfilaria. So he noted there's millions. There's like a lot. There's a high quantity of these microfilaria. That was his sort of first, not one of his big four, but that was his sort of logical assumption. And the next thing he thought, this is just a thought experiment, he thought if all these microfilaria became adults, there would be so much microfilaria in the human body that like the human would literally die. The mass of the human would literally be completely made up of adult microfilaria, okay? So he knew that this was not happening. He knew that these microfilaria were not becoming adults in the human body, because if that happened, obviously people would literally like die. And you don't die from elephantiasis, you don't die from filariasis. It just sort of destroys your body over a long period of time. So he knew this was not happening. And yet, that sort of made him question like, well, when do the microfilaria, so his sort of next logical question was, when do the microfilaria become adults? Like, where does that happen? And so he proposed the thought that maybe this happens somewhere else. So that's one of his first big contribution was maybe the microfilaria become adults somewhere else, okay? And he had, okay, so this is during the British Empire, and he had servants, and his servants slept outside in like a shack, okay? And he would come out at night. His servants had the elephantiasis. Okay, so here's the elephantiasis. And they would sleep in this shack. And he kind of had the hypothesis that maybe insects were related to this. So he would come out at night when the mosquitoes were feeding on servants, and he would collect the mosquitoes. And the first thing that he did is he would take the mosquito, okay, and he would take out its gut. He'd dissect out its gut, look at it under the microscope, and what do you think he's going to be looking for? The nematodes, the worms. And so he was the first person to see nematodes inside the mosquito that had fed on somebody who had filarial worms. So then he was sort of, so that's what sort of came to this hypothesis, that his hypothesis, you have to be careful how you word this. His hypothesis was that filarial nematodes become adults in mosquitoes. That was his hypothesis. His hypothesis was not that mosquitoes spread filarial worms. And actually, when we sort of figured that out, he did not believe that, okay? What he thought was the mosquito takes a blood meal from people who are infected with these worms. It flies to lay its eggs in the water. It lays its eggs and it dies, okay? And he thought it dies, the mosquito dies in the water and spills all the worms out into the water. And then he thought, okay, the transmission happens by somebody drinking or consuming some of this water, which has the nematodes in it, and then the cycle repeats. That's what he thought. So he did not think that the mosquito was actually a vector of the disease. That's what he thought at this time. And it's actually interesting. He always has his own thoughts. He had actually data to support this model, okay? So his data to support this model was if he took a mosquito that had worms inside of it and he sort of dissected it out and added water, he noticed that when the worms got into the water, they would start like beating their tails and swimming. Okay, so his data to support this idea of transmission through the water was that if you add water to the microfilaria, they start swimming. Okay, so that's what sort of his evidence, his logic behind why he thought this. This is not a dumb person. This is just a person who completely follows his own empirical experiments and develops what he thinks from literally his own knowledge. Okay. Oh, and just so that you know, the mosquitoes that he was collecting outside that shack were Culex pipiens. This is the southern house mosquito. Culex pipiens are famous for spreading filarial nematodes. Okay. Let's see. Okay, so then there's this person, Ronald Ross, and you're going to see how all these stories sort of intermingle with each other. Ronald Ross at the time was a person who was studying mosquitoes and malaria. And Ronald Ross was talking to Patrick Manson. They were sharing letters and communicating with each other. Okay. And so remember what I said about how at this time we did not even know about like the life cycle of mosquitoes. We did not even know at this point that a mosquito could take multiple blood meals. So this guy, Ronald Ross, just does the very, very first basic biology experiments on mosquitoes in the sense that he's culturing them, rearing them in cages. And he actually figures out that what Patrick Manson thought, that the mosquito just goes to the water and then just immediately dies. Ronald Ross kind of figured out that that does not happen. What happens is the mosquito lays eggs and then it seeks out another blood meal, and they can take iterative blood meals and lay iterative batches of eggs. Okay, so Ronald Ross figured this out. And once this was figured out, that sort of opened up the possibility that a mosquito could spread a disease. Because if the mosquito, we talked about this in the very first lecture, one of the key things is mosquitoes take multiple blood meals, and that's why they're spreading diseases, because they're taking multiple blood meals. But this wasn't figured out like until this person existed on the earth and did the experiments to discover that mosquitoes feed multiple times. And this guy, let's see, hang on. We went over this. So this guy will come up later in the malaria lectures. But know that they're talking to each other, and this is a very, very famous person who's sort of evaluating basic mosquito biology. Okay. Now there's another guy who sort of fits into the storyline. There's this guy, Bancroft. And Bancroft figured out that the worms move around in the mosquito. So he figured, so Bancroft found that the worms move in the mosquito. So Patrick Manson was the first one to see them in the gut, but Bancroft found them in other parts of the mosquito, and he found them in the proboscis. So he found them in proboscis. And as soon as you find the worms in the proboscis, right, what's this guy going to think? It's transferred during feeding, right? And so that's what Bancroft is famous for. His theory was that the worms might get into the mouth parts and get transmitted through the mosquitoes. And like I said, Patrick Manson did not believe this at the time because his whole hypothesis was that they're transmitted through the water. So he was not a big believer in this hypothesis until much, much later when it was very sort of rigorously proven. But Bancroft was the first guy to kind of posit this because he saw the Microfilaria in the proboscis. We went over this. Okay. So let's talk about Patrick Manson's second big contribution. So there was a unique thing with filarial worms. If you had a person who was infected with the worms and you sampled blood and you looked for the worms, depending on the time at which you sampled, the worms would either be in the blood or not there at all. So there was this strange phenotype where if you sampled from blood at night, you would see the worms. If you sampled from blood in the day, you took the blood sample in the day, you would not see any worms. So Patrick Manson observed this. So he's the first person to note that the worms, the Microfilaria, are nocturnal. You do not see them swimming around in the blood during the day. Why do you think this is? Why would this behavior evolve? That's pretty close. What connect? They're moving around. What's happening to them when they're moving? No. Okay, so if you're thinking that, that's the wrong thought. So the question is, so this is a phenotype that's exhibited by the worms, the Microfilaria. Why would the worms want to be in the blood at night, but not in the blood in the day? No, they do want them to be taken out because that's how. So you kind of got it, but your logic was opposite. You'll see that, and this is a pattern in vector biology that we'll see countless times. Is the pathogens evolve such that they can complete their life cycle, and to complete their life cycle, the Microfilaria need to get inside of a mosquito. So do they want to be cycling around in the blood when mosquitoes are not feeding? No. So what they do is they have evolved to cycle and swim around in the blood when it's most likely that the mosquitoes are feeding, okay? So, and the culex were in this area where the night feeders, so it's fascinating that the worms learn through evolution to be active when the mosquitoes are active. They sort of synchronize themselves such that they can be picked up, okay? Okay, so his second big contribution was that he discovered that these are nocturnal. Next slide. Okay, his third big contribution, Patrick Manson's third big contribution, was he knew at this point that mosquitoes are necessary for the worms to complete their life cycle. So he was the first person to posit if we kill the mosquitoes, then we can prevent transmission of the disease. Now, he did not even know, so again, to just reiterate, he did not even know that the mosquito transmits the disease. He just knew that the mosquito was necessary for the microfilaria to complete their life cycle, okay? So he just thought, if we can kill the mosquito, we can reduce transmission of this disease. So he was the first person to posit vector control. Vector control as a means of controlling disease. So that means, like, in order to control filariasis or in order to control malaria, you kill the vector, you kill the mosquito. And so this was like a big, I have a picture of here of the Big Bang. This was like a big explosion, a new thought occurring on the earth, the idea that we can limit transmission of pathogens by killing the thing that spreads them. This was like the first time somebody had had that thought on the earth. So he won a Nobel Prize because this was literally, and this was almost like right in the early beginning stages of germ theory. And this was like a monumental sort of thought that Patrick Manson had. Okay. And again, Manson and Ronald Ross, who was studying malaria, and Ross was studying mosquitoes, were communicating with each other. They were writing letters, okay? And Manson, here's Manson's fourth big contribution. Hang on, I'm looking. Manson suggested to Ross that maybe malaria is spread. So his fourth big contribution, oh, here we are, 4. He suggested to Ross that maybe if filariasis is kind of hanging out in mosquitoes, maybe malaria is also transmitted. Well, he didn't know filariasis was transmitted, but he suggested that malaria might be transmitted, or it might have some kind of a stage in the mosquito to complete its life cycle. Okay, he was the first person to suggest that, and he was suggested that to Ronald Ross, who then later discovered the transmission cycle of malaria. So those are his 4 big contributions to vector biology. So everything we sort of know, and even all much germ theory later, is sort of exploding because of these advances in the late 1800s by Patrick Manson. And then when he, let's see, yep, got that. And he was also communicating with Joseph Bancroft. And then what he did when he retired is he went back to London and he founded the London School of Hygiene and Tropical Medicine. This is still today a very, very good school for vector biology. And he wrote the first book on tropical medicine. If you hear quote unquote tropical medicine, that's essentially like vector biology to an extent. Right, like there's other things in the tropics, but vector biology is inherently in tropical medicine. So he wrote the first textbook as well. Okay, let's talk more about Bancroft. Bancroft was the first person to actually suggest that filariasis was transmitted by the mosquitoes, and that's because he saw the microfilaria in the proboscis. So I have just here a timeline. I've kind of talked about it, so I won't go through it. You guys can look at these slides if you want. Timeline of important things that happen. So that's like the history of filariasis. Let's talk about the life cycle of the worms. So you're going to see many of these pictures. These are life cycle pictures in this class. And they often involve the human, and they often involve the vector, and they often involve a reservoir. In this case, there's no reservoir because the human itself is the reservoir. But what happens is a mosquito will start here. Well, it's kind of like actually here. Let's start here at 4. What happens is a mosquito takes a blood meal from a human that's already infected with filarial nematodes. Okay, in the mosquito's blood, then in the gut are these worms, which gets picked up right here. Okay, now these worms are baby worms. These are microfilaria. So they go through 3 molts, or I guess it'd be 1, 2, it might be 2 molts because you get the 3rd L3 larvae. It's probably 3 molts to get the L3. But they go through about 3 molts in the mosquito itself. And while they're in the mosquito, they don't just stay in the gut, they get out of the gut and they go to the head and they go all the way down into the proboscis and they infect the proboscis as L3 larvae, okay? So one important thing here is they're not completing their sexual life stage in the mosquito. They're molting. They're molting in the mosquito, okay? So they're growing, they're feeding, they're shedding their exocuticle, which is the molt, becoming a later instar in terms of insect terms, but I guess these are not insects, so we just call them, I guess, larvae, okay? So the L3 larvae are what get into the mosquito head, and then when the mosquito feeds again, it injects the L3 larvae into the skin of the human, of a new human, okay? And then in the human stages is when the nematodes complete their sexual lifestyle, okay? So what happens is, if you have, oh, I guess I have a slide for this. In the human body, there's a system called the lymph system. The lymph system is used for, as the function of the circulatory system, blood, is to deliver nutrients to the cells of the body, okay? So the circulatory system cycles through the nutrients in the blood, and the nutrients come out into the extracellular fluid and feed the cells. Now, once the extracellular fluid has been depleted of nutrients, it needs to be recycled back into the circulatory system. So the way that that happens is through the lymphatic system, okay? So the lymphatic system, its function is to collect the filtered blood plasma, okay? So this is like the liquid that's cycling around in your body. It collects the blood plasma and it pumps it through the lymph system and it recycles it back into the circulatory system to be reloaded with nutrients to be then redelivered to the cells, okay? And you have lymph nodes. And what happens when you get bit with these worms is the L3 larvae crawl to your lymph nodes, these little nodule things, okay? They crawl to your lymph nodes. And once they're in your lymph nodes, they find mates. So you need a male and you need a female. And once they find mates, they mate. They become adults, or perhaps they become adults first and then mate. That's probably more likely. And then what they literally do, this is the most fascinating thing, they literally just sit in your lymph nodes for years and years and years. Some of the lifespan of these nematodes is 10 to 15 years, okay? And each female can lay millions and millions and millions of eggs. So they literally just sit in your lymph nodes and for 15 years continuously load your body with microfilaria that hatch, okay? So it's not the adults that are causing the disease pathology. It's the babies that they're laying in such sheer quantities that are loading your system. Got that. Okay. Went through this. There's different types of filarial worms. Many mosquitoes spread these. So this was the Mosquito Taxonomy. Many different mosquitoes in this tree spread filarial worms. So many. Anopheles spread these, Culex spread these, Aedes spread these. So these can be spread in general by many different mosquitoes. And there's two terms, this is a dumb slide, but there's two terms here you want to know, a patent infection and a latent infection. Okay, so some people are naturally like immune to the microfilaria. Some people can naturally just kill the microfilaria. So if you have adults but no microfilaria, that is a patent infection. Nope, that's a latent infection. And if you have both the adults plus the microfilaria, this is a patent infection. Okay, so there's a big diversity in terms of like resistance to the microfilaria that humans exhibit. What happens when you get this disease? So this is a nasty disease because it's not just like it's not just sort of making life uncomfortable, but it also sort of ostracizes people. People don't want to be around these disabled people who have these. And they're also, you obviously know that they're like a reservoir. What's up?
What like gives us the ability to like have some resistance to the...
Good question. We don't know. We don't know. If we knew that, then we could solve all the microfilaria. That would be a question for research. It probably has to do with the immune system. Certain humans probably have immune antibodies or something that can recognize the microfilaria and kill them. But we don't quite know that. At least I don't. I'm sure it's an area of active research. And the way that this happens, the elephantiasis, is, again, it's not actually the adults that are blocking your lymphatic system. It's them laying eggs, and the microfilaria, there's so many of them, they die, and they're made of like cuticle, right? Like they have like a hard shell, and their dead bodies accumulate in the lymph system and literally block the tubes. So what you see in elephantiasis is an accumulation of extracellular fluid that cannot get out because all the tubes are blocked by dead bodies of the microfilaria. And again, this is not something that just occurs overnight. You get this disease by having to live in endemic. Are you sleeping? Are you fucking asleep? Oh my God, he's like, really, are you sleeping? Okay. You'll have to rewatch the lecture. If you don't feel good, dude, you might have coronavirus. Get out of here. Okay. I might have to edit this. Okay. Back to my thoughts. It's a very slow disease. Over time, you get severe disfigurement. It happens over a long time. Let's look at the epidemiology. So the gray areas are the biggest one of these species is Wuchereria bancrofti. Its endemic regions are in the gray here. Okay, so it's all over the world. The dark here is a different species of nematode called Brugia malai. So bancroftii is really like the worst one. But we're getting, to be honest, like we're getting better at treating this disease. So 20 years ago, this is in 1974, so this is almost 50 years ago now. There were 250 million cases a year. In 1994, so 20 years ago, there were 106 million. So that we've already, in about 30 to 20 years, cut that amount in half. So we're getting better at dealing with this disease. There's new treatments, and it's reducing throughout the world. So I do think there will be a time at which we don't have this if the trend continues. Again, because this disease is extremely easy to treat. This is one of the vector-borne diseases that is extremely easy to treat and extremely easy to prevent. So I think with the improvement of the earth, we hope that this disease will be gone eventually. How do they diagnose it? So they just take a patient, filter out the blood. You can literally just run the blood through a centrifuge, and then you can just look for the microfilaria, look for the worms. You can also test antibody tests. How do we treat this? So this is fascinating. We treat this with ivermectin, and somewhere here, these slides are terrible. Like I have so many words. This is when I didn't quite know how to construct lectures, but somewhere it says 1 treatment of ivermectin per year is enough to completely eliminate or prevent microfilaria from accumulating. So if you have the disease and you just take one pill of ivermectin, it's either per month or per year, that literally prevents the pathogenesis. So it's a disease that's clearly curable. Ivermectin is this drug that targets, what is it, glutamate? It's like glutamate channels, glutamate gated chloride channels. So the theme with insecticides and many of these drugs If they're treating eukaryotes, it's usually they're inhibiting some kind of like neurotransmitter or neurological process with the neurons. So when you give ivermectin, it actually paralyzes the worms and it allows your body to sort of like clear them out more easily. Ivermectin, the discovery of it is really interesting. Actually, it was discovered on a Japanese golf course. Why do you think that would be? What do golf courses care about? Yes, golf courses care about turf grass. The biggest problem in turf grass is plant parasitic nematodes. So ivermectin was actually discovered by Japanese researchers on a Japanese golf course. And I would imagine that they were looking for treatments of how to kill golf course plant nematodes. And then once they sort of found like a basic chemical structure, this company Merck just sort of like optimized the chemistry to the point where they got ivermectin, the wonder drug. So again, like if we would just give people treatments of ivermectin, this disease would literally like go away. There's other things to treat it with. DEC, you don't need to memorize this. I'm just giving you like some of the options that people treat this with. DEC is a chemical that will inhibit it messes with the lipid bilayer in the nematodes. So there's other treatments. There's another drug called albendazole, which inhibits polymerization of worm tubulin. And I'll talk about this a little bit more. All these nematodes, these filarial nematodes, at least Wucheria, Bancroftii, Brugia, Malai, and Onchocerca ochengi, which we're going to talk about a little bit here at the end of the lecture, they all have Wolbachia. So Wolbachia is a bacteria we're going to talk a lot more in the semester. You guys already like know a lot about it. Wolbachia is mostly associated with insects, but it's also in these filarial nematodes. And in the filarial nematodes, the Wolbachia bacteria is an endosymbiotic bacteria that's essential for reproduction. So if you treat people who have filarial nematodes with antibiotics like doxycycline, the antibiotics will kill the Wolbachia and make it impossible for the filarial nematodes to reproduce. Okay, so there's a program called Anti-Wolbachia Consortium, AWOL, and they are developing drugs, new drugs that treat and kill Wolbachia selectively, because then that has very improved health benefits for people who have filarial nematodes by killing the symbionts. So that's really cool. Okay, quick. There's other, we don't have much time left, but there are other filarial diseases, okay? So there is also a filarial worm called Onchocerca volvulus, and there's also Onchocerca ochengi, so this is Onchocerciasis, these two different worms that cause this. This is called river blindness. So these worms are similar, but a little bit different. They form, the adults form these nodules underneath the skin, and the same difference, these adults lay many eggs, and the microfilaria crawl through your eyeballs and make you blind. So when it's actually the physical act of the microfilaria crawling through your eyeballs that destroys your eyes. This is river blindness. The reason it's called a river blindness is because it's associated with rivers. What lives in the rivers? Black flies. Black flies are kind of like taxonomically kind of similar to mosquitoes. Similar concept in that sense that they're aquatic. They have aquatic larval stages and aquatic pupal stage. They're telmophages, so they're not like mosquitoes in the sense that they're not selenophages. They're not sucking your blood by a tube that goes into a capillary. They have sharp mouthpieces that slash your skin, create a pool of water, or a pool of blood that then the fly sucks up. And these are the creatures that are spreading onchocerciasis, river blindness. And just so that you're clear, this is not to be confused with Loa loa, which is another filarial nematode where the adult actually like lives in your eyeball. So these are different, but you can have both sometimes. If you live in a region that has both Loa loa and onchocerciasis, you can get both. So this causes blindness in people. Let's end it there. Any questions? Okay. Was it that boring? What is that?